Anyways, my name is Brett Augsburger from Auburn University. I'm very happy to be here today. So I've got 15 minutes faced with the diminutive task of explaining how non-equilibrium thermodynamics and evolution work together to explain, possibly, why organisms age. So I'll get right into it. So if we were to ask an evolutionary biologist the question of why organisms age, it's likely we'll get an answer, something along this lines, that organisms age, or most organisms age, because configurations that maximize fitness tend to produce organisms that are not invulnerable to aging, which is sort of a circular answer, but an alternative answer is that organisms are at a fitness island which does not produce an immunity to aging, but there could be a fitness island that we just haven't been able to reach because of the fact that it's too far away from our current fitness island. So on a fitness landscape, we just haven't been able to get there. So granted, this is not a very satisfactory explanation of why we age, but I think it's important to keep in mind with any theory of aging, the evolutionary considerations. So why do inanimate objects age? Well, I would argue that this is best explained by the second law. The fact that inanimate objects, they're structured, and with that structure comes the fact that they're in non-equilibrium, so they have thermodynamic potentials. The second law of thermodynamics stipulates that they're going to produce internal entropy. The produced internal entropy is going to be degradative. You can have irreversible processes that are effectively going to degrade. In this case, I'm not sure which configuration is the more desirable, but that's what happens with cars eventually. So how is this relevant to aging? And well, first of all, a lot of the arguments, several arguments have been used to discredit thermodynamics in aging. There's kind of a ongoing prejudice, it seems, against thermodynamic principles in aging. And two of the main arguments are, the first one being that the second law only applies to isolated systems. Of course, this is really predicated on a out-of-date interpretation of the second law. The more modern interpretation, of course, can be utilized towards any type of systems, and the second law is universally applicable. So the second thing, the second argument you hear is that organisms are open systems, therefore that they combat entropy increases by utilizing energy and mass transfer. Of course, this is true, but you can't ignore the fact that there is a potential impact of these long-term persistent degradation from these thermodynamic forces. So often you hear biological deterioration, degradation mentioned, but they're really rarely explained as far as what's really behind that. So as far as thermodynamically, how would we describe organisms? Well, they're highly ordered. They're non-equilibrium dissipative structures, so they're going to produce degradative internal entropy, just like a inanimate object. And yes, of course, they are going to exchange energy and matter with the environment. So if we just wanted to look specifically at the degradation forces, we could create a system like this. This is just hypothetical, so this is going to be kind of a theoretical exercise here. So if we assume, in this case, we're going to take a fixed volume of cytosol, and we're going to look at a single biomolecule. We're going to focus on a single biomolecule, although we're going to have everything there, of course, all the other biomolecules you would have in a cytosol. We're going to take the temperature and pressure, and they're going to be in equilibrium with the surroundings. Concentrations will be of everything aside from our biomolecular interest. We'll hold it constant with a chemiostat, and we'll maintain a thermomechanical equilibrium, of course not a chemical equilibrium, and we're only going to look at the reactions involving our biomolecule of interest. So basically we're going to look at how a biomolecule would degrade. And we also have to inhibit biomolecular repair or replacement. So what we'll see is initially we'll be in, of course, non-equilibrium, and we're going to transition through a very, very large number of non-equilibrium states, and we'll have reactions between our molecules. So basically, these are going to be unintentional reactions. These are going to be the reactions that we don't want. They could be ROS, cross reactions with other components. So what we'll have is a large number of these reactions occurring at different rates. We're going to create internal entropy. We can predict using this equation if we know everything. And basically, we're going to produce entropy until we reach equilibrium. And at equilibrium, all our reaction velocities and reaction affinities will reach 0. The total entropy produced during that time can be calculated with this formula. And if we know the total entropy produced, taking it all the way up to equilibrium, we can generate a new parameter that I've called the degradation state to basically describe any intermediate degradation state of the population of biomolecules. So that would be calculated by taking the entropy increase at that time and dividing by the total entropy increase when you reach equilibrium. So to maintain a steady state, what is required, of course, is to satisfy our entropy balance. And we know that we're going to be producing entropy, so we need to counteract that. And the way that we can counteract that is by using a replacement mechanism. So let's suppose our system incorporates a replacement mechanism that replaces nRC moles per second of degraded biomolecule with new biomolecule. This steady state can be maintained if the bottom equation is satisfied. So now if we make one more assumption, and that is that our replacement mechanism is not discriminate to the degradation state of the biomolecule, then we can use this equation. And so what we see, as what I'm showing in the figure here, is that the replacement rate required to maintain a particular degradation state is inversely proportional to the degradation state. And this kind of perceives what we, or it demonstrates what we can perceive intuitively, and that's that attaining a perfect fidelity state would require an infinite replacement rate. And I should say on the last slide that if certainly improving the damage recognition of the replacement mechanism so it is more biased towards replacing the most damage, that will shift the curve to the left, but the trend is still going to be the same. So basically, the idea is it's impossible to have a biomolecular pool that's in a state of perfect fidelity. So I suggest that any biomolecule needs to be capable of withstanding some amount of degradation and still perform its biological function, whether it be slight oxidation or what have you. After a certain amount of damage, certainly the biomolecular performance will decrease, and it would be expected that this rate would be initially increasing. So I think we can represent the performance by a logistic curve as shown with P. So the graph on the right demonstrates or shows a representation of a general relation between biomolecular performance and degradation state. And the thing I think is important here is, and this is a concept that I haven't tested this, it's just an idea I have, is we think about accumulated damage and we've got to fix damage as it accumulates. And I think it's probably more likely that it's more a degradation state issue. And if we can maintain repair and replacement at a certain rate, then we'll maintain the performance, so we're up at least towards the top of the curve in blue. If the repair and replacement is too slow, then it's not that we really accumulate, it's more that the quality of the molecules have decreased. Now, there are some exceptions. There are definitely some biomolecules that will accumulate. But I think they're more the exception than the norm. So how do we manage internal entropy production in an organism? To protect against the loss of homeostasis, we need to be able to balance internal entropy against causing the degradation. So there's two main ways we can do this. The first is produce negative entropy to offset those effects of the degradative internal entropy, and the second is to minimize the degradative internal entropy production itself. So there's been a lot of talk about A. Most focus is on A, and that would include expression systems, molecular chaperones, proteasomes, etc. But item B has been generally overlooked. And this factor is proportional to some of the contributions from all the thermodynamic potentials, including chemical reactions, heat, mass, transfer, other effects. These potentials depend on the magnitude of the respective force and the ability of the organism's molecular structure to resist the force. So a biomolecule could be optimized to function at a specific, what I defined earlier, degradation state and resist degradation to varying degrees. But this has to be weighed against other performance, other factors such as catalytic activity, energy utilization, substrate specificity, and resources for production, repair, and replacement. So if we view the aging as a progression through many different steady states, and then let me define that real quick. When I say steady state, I mean if you compare us today to us in a week, we degrade as far as degradation, we don't really look that different. 10 years out, yes, we're different, but so that's what I mean by steady state. So it's kind of like we're at one degradation state for a period of time, and things are slowly transitioning. And the question is, why do we transition from these states? Well, there's two classifications of components that I think are particularly vulnerable to permanent damage. The first is those that lack capability to be renewed, and the second would be information containing biomolecules, those that encode for other molecules, and of course that would be DNA. So I'm going to skip the first one because in interest of time, and I'm going to talk about how the second one might be linked to internal entropy production. So of course, DNA molecules contain the information that encodes for all other classes of biomolecules. We all know this. Contains instructions for cellular processes. So it's clearly its DNA integrity is very important. And to preserve the integrity, we must the DNA must be repaired perfectly so the information is intact, or the molecule must be eliminated from the molecular pool. So if we could define, or not define, rather, we could calculate the rate of mutual DNA information loss, use this equation. So we have a block for replication on the right. We have static conditions on the left. We have static noise, that's basically your mutation during non-replicating conditions. We have mutation detection, and then we have replication noise and mutation detection during replication. Obviously, mutation during replication is usually a much bigger problem. Okay, so I'm going to claim that perfect DNA repair is a myth. And the reason for that is that in a cell, you only have a single level of redundancy as far as DNA information is concerned. So if you lose a base on one strand and you have suffered an insult to the other strand before the first strand is repaired, you've lost information. So it's not really a matter of whether or not you can maintain perfect DNA repair. It's how long you prevent mutation. So higher levels of DNA repair enzymes also, I believe there's diminishing returns because of this factor, because of what I mentioned earlier about the performance of the molecules when they're expressed and enzyme kinetics. So the germline's going to mutate, but what it must not do is mutate so much that the offspring have lost fitness. So basically, in terms of increasing priority, the somatic cells can mutate a certain amount, which can be more than what the stem cells mutate, which could be more than what germ cells mutate. So this puts different priority levels on maintaining the information and integrity of those molecular pools. So mitochondrial DNA are a special case because mitochondria range from several hundred to thousands per cell, and every cell contains, or each mitochondria contains mitochondrial DNA. Dysfunction has negative downstream effects on every biochemical reaction that requires ATP. And the mitochondrial genome is more susceptible to mutation. As integrity is compromised, the production efficiency will diminish, and this is going to lead to downregulation of cellular processes. Mitochondrial numbers are maintained by fission-fusion, or quality rather is maintained by fusion-fission and mitophagy and mitobiogenesis. So you're going to have a high rate of mitochondrial replication. And we know that the mitochondrial replication rates vary considerably between species. Lower replication rates lead to lower quality mitochondrial components and reduce energy production efficiency, but the rate of mitochondrial DNA integrity loss will be less. Conversely, higher replication rates produce higher quality mitochondria, enhance peak energy output, but increase the rate of integrity loss. So this suggests there's likely to be an optimal replication rate that would balance the rate of mitochondrial DNA integrity loss with an organism's energetic requirements. Okay, so attempting to put it all together, living organisms are non-equilibrium systems that constantly produce internal entropy and undergo biomolecular degradation. So this degradation must be countered to prevent structure from deteriorating. No population of biomolecules is in a perfect state, and the quality of the biomolecular component is going to be determined by the rate at which the molecules are replaced and damage and repair. The quality also depends on the ability of the biomolecule to resist thermodynamic potentials and the magnitude of the potentials themselves. The quality requirements contribute additionally towards overall metabolic rate. So all these parameters may vary considerably between organisms, and the rate of loss of DNA integrity then can depend on factors that are specified by the organism's genetic program. The nuclear and mitochondrial matrix microenvironment conditions will affect the rate of molecular insult on the nuclear DNA and the mitochondrial DNA, metabolic, athletic. Additional factors can influence the optimal microenvironment conditions that maximize fitness. Then back to the mitochondrial argument, higher quality mitochondria provide better power densities, so this could suggest that organisms with higher general biomolecular quality requirements and higher metabolism would be more likely to operate with higher mitochondrial replication rates. This would increase the rate of mitochondrial DNA integrity loss and shorten lifespan. On the other hand, organisms with low metabolic rates and those with low biomolecular quality requirements would tend to have lower mitochondrial replication rates. So, sum up real quick. I'm saying that most organisms and indefinite preservation of fidelity isn't a strategy that maximizes fitness, so thus most organisms are susceptible to aging, and what we see is this downward cascade as things get worse and information is lost. And that's about all I have.
This is very interesting, but one thing that got me a little bit confused is your assumption about unbiased molecular replacements.
Yeah.
How sensitive is your results to that assumption? Because I think we know that damaged cells commit suicide basically, and that's pretty biased. And whenever you have DNA damage, the cell has very nice mechanisms to fix particularly that damage without replacing the whole DNA.
Right.
So I'm not sure if that's a fair assumption, and if you could tell us maybe how sensitive your outcomes are.
Certainly, I should have mentioned, that's not going to apply to DNA. So DNA is totally different. I was more looking at proteins, lipids, things like that.
So very microscopic.
Right, but on the molecular level, and there is going to be some bias. There's more equations I haven't put in there, and I've been working on the equations that take the bias into account. And I've concluded that everything kind of remains the same, it just gets shifted, but I haven't completed all the calculations yet.
I want to kind of warn you against this perception that the cell has this, as you said, nice mechanism of annihilating itself. The role of apoptosis in our physiology and repair mechanism, especially in adults, is enormously exaggerated in literature. There are only very few tissues which are prone to apoptosis, while the vast majority of body, you can never induce apoptosis in the cells. So the apoptosis based on DNA damage is started in lymphoid system and blood, hair follicles, and 3 layers of cells in small intestine. That's it. In all other tissues, DNA repair is occurring.
That's still pretty good. I mean,
yeah, it's about 1% of your body weight. I agree.